What a start to things. Anyway, I hope this video trailer provides some context for you and helps you as you go through the full slideshow. But I want you to go through this lesson on lipids. I'll touch on it very briefly in class. But with our big idea that chemistry drives the biology, I want to focus on a couple things and a couple highlights for you. Specifically, the structure of triglycerides and phospholipids and how that chemistry impacts the function that it has. And so I'm going to go through this really quickly. Hopefully this trailer is of benefit to you. It's important to recognize what the functions of lipids in our bodies are. Now, lipids by definition are things in our bodies that are non-polar. And so there's different types of lipids. We'll look at those, and we want to focus specifically on the triglycerides and phospholipids. But lipids have these 5 general functions in our body as long-term energy storage. That's how we store long-term energy. They provide insulation, protection of our organs. They act as hormones or chemical signals, and they perform the backbone, or are composed of the backbone of our cell membranes, which we'll pick up on. So those functions are important, but what determines whether fats are good for us or bad for us? What are the different types of fats? That's what we want to get into in this lesson. So the general classification for lipids, as we look at it, there's 4 types, and we're going to focus on fats or triglycerides and phospholipids. Steroids and waxes act as chemical hormones typically, or as barriers to different surfaces. We're not really going to get into the chemistry as much on those. So for the first 2 examples there, we really want to focus in on what is the specific chemistry and how does that impact its biology. Okay, so when you hear the term fats, what we're really talking about are triglycerides. Triglycerides are made up of 2 parts. There's a glycerol molecule, and that glycerol molecule, if I bring it in here, that glycerol molecule is a 3-carbon molecule, and it has hydroxyls on the side. So that is a polar molecule, but it's composed of then 3 fatty acids that come together in that. And so our fatty acid chains look like this. I'll bring in, there's 3 here, okay? And these 3 fatty acid chains, you'll notice that there's a carboxyl on the sides. So we have this carboxyl and a hydrocarbon backbone along there, and a hydrocarbon backbone, and then this carboxyl group. And so when a triglyceride comes together or a fat comes together, it has a very specific structure. This is not a polymer. We don't just keep adding fatty acids onto it. And when it comes together, it's held together by an ester linkage. And so an ester linkage is this condensation reaction that's going to occur between the hydroxyl and that the other hydroxyl from the carboxyl group. And so a water is going to be, oh, you see my left hand there. Okay, a water is going to be taken out of here, and we'll be left with an oxygen there held in between. Okay, and so if you're looking at a condensation reaction, you can show how those 2 hydroxyls come together and an oxygen is going to be left behind. But the basic structure for a triglyceride is that glycerol molecule, another GLO word, and then 3 fatty acids. So this diagram that's in your notes, this might be a more useful diagram for you. What I want you to focus in on here is this condensation reaction that occurs as you have the hydroxyls from the glycerol and the hydroxyl from this carboxyl group. It's a bit hard to see in this diagram, but this is a double bonded O. It's a bit hard to see. Okay, that is a double bonded O. And so we see here with each one of these, we have 3 water molecules that are given off in that condensation reaction. And overall, this creates this triglyceride molecule, and we have now this covalently bonded oxygen holding the fatty acid chain to the glycerol molecule. And this linkage, this is referred to as an ester linkage. Okay, so we had glycosidic linkages hold carbohydrates together, ester linkages hold these triglycerides together. So I'll show you some examples in our next slide, but I want you to get the idea that when we look at triglycerides and we want to know are fats good for us or bad for us, it is based entirely on the unique set of fatty acids that make it up. There's 3 fatty acids in a triglyceride, and those fatty acids can be different. They could be short, medium, or long, and the chemistry of that will change its biology. Those fatty acids can be saturated or unsaturated. Again, the chemistry changes the biology. The length of that chain, the number of bonds, single or double bonds, ultimately determine the specific properties of that fat and determine whether that fat is good for us or bad for us. And so I'm going to highlight that in our next slides. Okay, so we talk about different types of fats. When we talk about fats, we're really talking about triglycerides, all right? And so saturated triglycerides or saturated fats versus unsaturated fats, we're going to take a look at the chemistry here. And when I pull this image up to look at the chemistry, if we're looking at saturated fats have all single bonds along this hydrocarbon backbone. Here's my carboxyl group on this side, okay? And then it's all single bonds, and so these are linear straight chains. And when they come together in a triglyceride, they're going to have unique properties. And you can go over the note for those properties, but they're going to be held together much stronger versus an unsaturated triglyceride, where now unsaturated means we have a double bonded here between a couple carbons. And that double bond is often going to create a kink or a change. And so it's not super obvious in that first diagram, and I'll come to that in a moment. But here you can see this double bond. It's going to create that this fatty acid, the shape has changed. It's not just a straight linear chain, but it's going to have a bend to it or a bit of a kink. And that's going to mean in that bend configuration, the bonds that hold that triglyceride aren't as strong. So these unsaturated fats exist as oils, and in our body they have different characteristics versus a solid saturated fat, which would be like the butter in your cupboard that's solid. Now, as we look at this, we can also see the difference between a trans fat and a cis fat. And when we look at trans fats versus cis fats, notice here, this is a small change of how the chemistry drives biology. But in a trans fat, the hydrogens are on the opposite side. And when the hydrogens are on the opposite side, it means that this is going to stay in this linear straight chain. It's going to be hard to break down, and our bodies, our enzymes have a hard time dealing with this. So trans fats are held together very strongly. It's still double bonded though. It's still an unsaturated fat. So if you say, well, unsaturated fats are good for us, well, not trans fats. They're really bad for us, okay? Because they hold together really tightly. A naturally occurring unsaturated fat, cis, this is the naturally occurring unsaturated fats that exist. Notice here that the hydrogens are on the same side. That small change in chemistry means that this shape is going to be more of a bent structure. And so the cis fat will have different characteristics than the trans fat, which has different characteristics. The chemistry here drives the biology, okay? And it's all about the placement of that hydrogen or whether we have a double bond or single bonds. The final example I want to highlight for you is because sometimes we look at trans fats and say, well, they're not good for us. Well, it depends on a few other things. It can also depend on, I brought the wrong graphic in here, it can also depend on whether we have short chain or long chain or medium chain fatty acids. And so if you think of coconut oil has a lot of health benefits for us. Coconut oil is a medium chain fatty acid. So not only does the type of bond matter, the placement of that bond can matter too. I'll explain that in a moment. But the length of that fatty acid chain will affect our ability to digest it, break it down, use it for energy in our cells. So medium chain fatty acids, like a coconut oil, are really good for us, okay? The final example I want to highlight, because you've, I'm sure, heard of it, is omega fats. Omega-3 fats versus omega-6 fats, you sometimes hear about those things. And when we talk about omega fats, all we're talking about, if we talk about an omega-3 fat, chemists like to number their carbons. So if this was carbon 1 here by the omega end, carbon 2, carbon 3, an omega-3 fat means that it has a double bond between carbon 3 and carbon 4. Okay? If we change that double bond, if we changed it to between carbon 6 and carbon 7, if the double bond was there, it'd be an omega-6 fat. So when we look at these fats, are they good for us or bad for us? It depends on the length of the chain, the double bond or not, where that double bond is located, okay? The hydrogens on either side of those double bonds, it is all due to the fatty acid chain, whether a fat is good for us or bad for us, how it's gonna react. So again, chemistry drives the biology. It's entirely based on that fatty acid. The final thing I want to highlight for you is, what if we change the triglyceride? What if instead of a glycerol in 3 fatty acids, which has unique properties, What if we get rid of one of those fatty acid chains? And so a phospholipid gets rid of one of those fatty acid chains. We still have the glycerol molecule. We have now 2 fatty acids. And instead of a 3rd fatty acid, we've added a couple polar groups. Okay, there's a phosphate group on here and a nitrogen group on here. And so by adding a polar group, polar now is water-loving. And so that head we refer to as hydrophilic. It will seek water. It will orient itself out towards a water-based environment, whereas the nonpolar tails are hydrophobic. They hate water. And so when we look at this, this forms the backbone of our cell membrane. When we start to kind of draw a cell membrane, we would draw kind of the polar head, and then these fatty acid tails, they're going to orient themselves toward one another. And so again, the chemistry drives the biology. We now have a molecule that's polar on one side and non-polar on the other side. And those things aren't going to mix, and that's going to create the barrier for our cell membrane, which we'll talk about more in a moment. Okay, this cell membrane, we'll get into this more in our next lessons. But with this bilayer, we now have a barrier where water and other polar substances can't just pass through there whenever they want. We're going to ultimately construct a membrane that has different channels, different protein channels that are going to allow certain things to come in, certain things to go out, but it allows us to control our environment. And this is really the basis for all living things, to have a cell membrane. And that's based on the unique chemistry of that phospholipid. And that chemistry is just because we've removed one of the fatty acids. So again, this whole lesson, it's about the type of fatty acid, what is the chemistry of that fatty acid, and how does that impact us. I hope this helps. Good luck.